Hey everyone, welcome to the Basics lecture on cell cycle and mitosis. Cell division is a normal part of organismal life. It's responsible for growth, tissue repair, and even reproduction in some organisms. And when the cell cycle goes wrong, it's important because it can result in the uncontrolled cell growth that we see in cancer. Let's look at the cell cycle first. There are two main phases, interphase and mitosis. Interphase is divided into three subphases. In G1, the cell grows, makes proteins, and builds organelles. Some cells rarely or never divide and remain in G1 for the life of the organism. In S phase, the chromosomes become duplicated. And in G2 phase, cells finish growing and go into mitosis. During mitosis, the chromosomes separate and the cell divides into two daughter cells. Before we go any further, you need to be very familiar with some vocabulary associated with cell division. Here are the list of words I'd like you to look up now because we're going to use them repeatedly in the next section. These are well defined and only a little bit complicated, so go to chapter 12 of your book and read about them and define them and why they're different in your notes. Okay, the next two terms are often very confusing to students, so let's look at them more closely. A chromosome that is not about to divide is unduplicated. It's a normal double-stranded length of DNA. When a cell is preparing to divide, each chromosome has an exact copy made. The new copy remains attached to the old copy. Together, they are still just one chromosome, but it's called a duplicated chromosome and it contains twice as much DNA. Each strand is called a sister chromatid. In humans, before DNA replication, each somatic cell has 46 chromosomes. After DNA replication, each somatic cell still has only 46 chromosomes, but the chromosomes are duplicated. Now, let's talk about haploidy and diploidy. Imagine that we have a mother's egg that has only 3 chromosomes, one each of a red, a blue, and a green. Chromosomes aren't actually in different colors, but it helps in the cartoon form. Since they come from the mother, the chromosomes are called maternal chromosomes. The egg is haploid because it has only one copy of each chromosome type. Since it has 3 chromosomes, N is 3. A sperm has also 3 chromosomes, but may have slightly different versions of genes on these chromosomes. These are paternal chromosomes, and the sperm cell also has an N that equals 3. Now, when they come together, sperm and egg, their offspring have the chromosomes from both parents. It now has 2 copies of each type, 2 blue, 2 red, and 2 green. One is a paternal copy and one is a maternal copy. Since these chromosomes are very similar but not identical, they're called homologous chromosomes. Since there are 2 copies of every chromosome, the cell is diploid, or 2N equals X is the language we use to refer to a diploid organism. So take a minute, look at the difference between homologous chromosomes, maternal and paternal. Lastly, students struggle to understand the difference between homologous chromosomes and sister chromatids. So let's think about that. Here's our organism again with its 6 chromosomes. It's diploid. If this organism is going to divide, each of the chromosomes is going to duplicate. The cell after S phase still has just 6 chromosomes and still has a maternal version and a paternal version of each chromosome, pointing out the blue ones. But there are now homologous chromosomes, still, that hasn't changed from G1 to G2, but there are now sister chromatids for each chromosome. Okay, so take a moment here, look at the difference between sister chromatids and homologous chromosomes. Okay, we've added several additional terms to your vocabulary list, so take a minute to write out the definitions of each and make sure that you're comfortable with them. Haploid, diploid, maternal and paternal chromosomes, homologous chromosomes, sister chromatids, and DNA replication. Before we finish up this video with mitosis, let's look for a minute at condensation. As we saw in previous lectures, DNA is stored as nucleic acid strands wrapped around proteins in the nucleus. When a cell is preparing to divide, these loops are packed even more tightly and looped repeatedly to create a condensed chromosome that is visible under a light microscope. Here's an image from a computer, or here's a cartoon of these condensed chromosomes, and you can see that there are homologous chromosomes organized next to each other. These are cut and pasted by a computer. Chromosomes don't normally line up just like this. You can see the homologous, one's paternal, one's maternal, but each particular chromosome has sister chromatids attached to it. And humans have a total of 46 duplicated chromosomes and 23 homologous pairs. This particular image is called a karyotype. Sex chromosomes determine the sex of the individual. Chromosomes that are not associated with the organism's sex are called autosomes, and humans have 44 autosomal chromosomes and 2 sex chromosomes, or 2n equals 46. Finally, we've reached mitosis. You need to know what is going on at each phase at a basic level. Using the figure in Chapter 12, organize the following events under the correct phase. This is the information I want you to know for mitosis. Then read through the whole figure after you've filled out your notes and be comfortable with the order of events. Every duplicated chromosome has a centromere, which is an actual region on the chromosome where sister chromatids are most tightly attached. Kinetochore is a group of proteins that bind to the centromere when division begins. The kinetochore binds to the chromosome and then presents an outward binding site here and here, where spindle fibers attach, like so. Spindle fibers are microtubules with the minus ends at the poles and the growing plus ends reaching towards the chromosomes. Inside each kinetochore are dynein motors, shown in this cartoon. As soon as a microtubule binds to the kinetochore, the dynein begins to try to walk along it, pulling on the sister chromatid. But microtubules bind to the opposite side, here and here, are also pulling on the other direction. This has the effect of centering all the chromosomes in the center of the cell. During anaphase, the proteins holding the sister chromatids together are broken down and the centromere breaks, and the dynein is able to pull the sister chromatids to opposite ends of the cell. This completes our basics video for today. Write out the answers to the following questions to study for the basics quiz. Thanks for watching.